Hi everyone, welcome to another episode of my tutorials on Ontology for Bioinformatics. In today's video, we are going to look at how to perform a Go enrichment analysis. So by the end of this tutorial, you are going to generate this plot which is being shown right now. Now, for those who are beginners in this kind of analysis, the two letters GO, that's Go, they stand for Gene Ontology. So if you're a beginner, I will encourage you to check this page out. The link to this page is in the description box. So check this page out and then read the introduction here. We have the overview and introduction. So this will give you an overview of what the Go enrichment analysis is about. We are also going to use this to Go Seq. All right. So for now, just do the overview, read it, but don't worry about the analysis here. We are going to cover them later on. All right. So just check this and then read everything here. That will help you get started. All right. So for this tutorial, we are going to do everything on the Galaxy platform. All right. So we are still going to run this to Go Seq. We are going to run it on the Galaxy platform. All right. So don't worry. The example data is also available. So I'll show you everything. Now, this tutorial here is a continuation of this one. Let me just scroll up so that we see it well. The link to this page is also in the description box. So this is a reference-based RNA Seq data analysis tutorial. So I've covered this tutorial, I've covered some parts of it. So what I'm going to do is a continuation. So the Go enrichment analysis is, let me just scroll down. It comes after you have identified the differential expression. So I've covered all these steps up to this side here. So we are now continuing from this side. So this is where we are. So functional enrichment analysis of the Dagents, and we are doing the gene ontology enrichment analysis. That's what we are doing here, right? So I also encourage you to read this introduction here to help you get up to date or to help you get started when it comes to the Go Seq tool. So just make sure you do that. So let me also say that you can perform the gene ontology analysis after you have identified the differential expression. So if you want to learn how to identify differential expression, then please check the description box. I have tutorials that show you how to do that. So I've also covered that section in one of my videos. So just check the description where you find links to the videos that covers these initial stages, and then you can proceed. Okay, so let's do this. So let's go back to Galaxy and then we can start the exercise. So I want you to come to Galaxy and then first log in and create an account, and then you can proceed. I already have the example data, so we will look at how to get it and then run it. So don't worry. First of all, I want you to create a history. Come to this side. So click on this to create a history. Let me just do what's here. I'll just redo it again. So I'll just click on this plus to create a history, and then you have this coming up. So here you can edit and rename. So I'll just say Go enrichment analysis. You can use any name of your choice. I can also add today's date to it just to keep track of my activities. So the name is up to you, but just make sure I have a name that you can track, and then I will save it. So let's save it. All right, that has been done. All right. Now I want us to import the example data that we are going to use. I'll also briefly explain the data set, so don't worry about it. So go to this GitHub report of mine. There's a page, and I'll leave the link to this page in the description box. So when you are here, you'll see two files, gen IDs and lengths, and then gen IDs and differential expression. These are the two files we use. I'll talk about them later on. So we need to get the links to this files, the download links. So let's start with this one. So click it, and then you'll be sent to another page. Then you will see this. We have gen ID, we have length. So we have the gens and we have the gen lengths. So the gen lengths, you can get it if you run feature counts. And I have a tool on that. There's also a tool on Galaxy that can help you to get the gen lengths. It can compute it for you. So you get this, and then you get something like this, and then you can use that as one of the inputs. We need two inputs. So this is the first input. So let's get a link. So to get a link, come to this side raw. So right click and then go to copy link. that will get you the link to the file. You can also click on raw. So when you click on raw, you'll be sent to this page, and you can just copy this link, the download link. Right? So all those activities, all those approaches will give you the link. So you have two approaches to get a link to the file. Now let's go to Galaxy and then come to this side. So click on upload data. By the way, the Galaxy, you can use this one, use galaxy.org, or you can also use the EU one. Any of them is fine. All right. So click on upload. And then when you come here, click on paste/fetch data. So click it. And then when you are here, paste the link. And then with the type, let's select Tabular. So click it, and you can even type Tabular to show you like this. So click it, and that's it. And you can click on start. So that has been done. So once you click on start, you should see the file generated here. So you can close it. You are going to see that the background is gray. It means the job is in queue. So it's going to be imported very soon. So whilst we are waiting for this, let's also import the other one. So we'll go back to GitHub. Let's go to the main page. Let's get the link to this file as well. So the same approach. So click it. And when you click it, you have this coming up. So this one is gen IDs and differential expressions. So here we have the gens, and then we also have the status telling us whether it was differentially expressed or not. So these are the two inputs. So this is the second input. So here you see true, that means that this gen was differentially expressed. So we also have some that are false. Let's just scroll down a bit to see that we can see some of them here are false. So that means those were not differentially expressed. So you need something like this. So these are the two inputs that we need. Okay, so come to raw, right click, and then copy the link. Come to Galaxy, click on upload, come to paste/fetch data, come here and then paste it, and then click on start. So that also generates another file for you. You can close it. For the moment, both jobs are in queue. So you have to wait and then make sure the background turns green. I'm using this Galaxy platform, but you can also use the EU one, which is here. Let me just check. Okay, so I think this is one of my histories. I will just click on this one. Yeah, so you can also use the EU one, which is here. And then you can also import, or just import anytime, then we can proceed. So I realize that sometimes this EU one, the Galaxy EU one, I get my work done faster here because it seems there is less queue compared to this one. So you can decide if you want to use this or not. Anyway, I think I'll switch to this one so that I get things done faster. So I'll do that here. Okay, so the data has been imported, the files, we have two of them. Now you notice that the first file, this one here, when I was importing it, I set the type to tabular. So when you click it, you should see tabular here. So that will tell you it's tabular. Now for this one, when you click it, you may see this or not, all right? But if you don't see tabular, then what you can do is to come to this side, click, and then when you come to this side, data type, when you come here, click, and then type tabular. Then when you type tabular, it should come, and then you can select it. Right, that's it. So tabular, you click it, you can select it, and you can just save it. Right, so let's just do it. So tabular, let's just do it here. It's just tabular, and you just save it. So you should see it like this. All right, that's how we do it. Okay, so if you do it, if you should click on both files, let me just wait for it to load. If you click on both files, you should see the format tabular showing. Those are the things that you have to do. And then we can proceed. So I'm only showing you so that you'll be able to know how to change it at that. Because sometimes some of the Galaxy tools, if they don't see the formats that is supposed to see, then the tools may not work. So this is a way to show you how to change the file types. All right. So when you click, You should see Tabular here. When you click this one, you should see Tabular here. These are the requirements. All right. So now we are done. So let's click back to Galaxy. Let's go to the home page and then we can proceed. Okay. So we have our two inputs. Let me just click and then hide the information. So we have our two inputs. So now we are going to use GoSeq. This tool. GoSeq, that's what we are going to use. Actually, it's an R package. And so I'm kind of working on the R version of this tutorial. So once it's ready, you can check the question box. So you'll find the link to that video today, right? So that one will also be uploaded very soon so that I can have both approaches. So we are using GoSeq. So we require the gen IDs and differential expression. We also require the gen IDs and lengths. We have both files here. And so we need to run GoSeq. So come to the tools, type GoSeq. It will show up. So click it. And when you click it, you come to this parameter. So this one is differentially expressed gens file, which is this one. This one, yes, it has been selected. But if you don't see it, make sure to select it. Then the gen length file, it's this one. So you click and then you select it. And then after that, there are other options we have to specify. Now, the data I'm using is from the tutorial here. So I just put the tutorial and then I've got the example data. Right. So the data that I'm using, I followed all the tutorials here, followed the instructions, I got the Dagents, I did some further processing, and I got the file. So basically, we expect to get something similar to what will be shown here at the end of this video. So just make sure you are using the same file. All right. So if you have your own data tool and you want to generate some plots like that, you can always contact me and then we can have a session on that. My contact link is in the description box. Okay. So let's do this. So let's get back to GoSeq. So we have done that. And now when you take a look at the instruction, it says gen categories, get categories. So we come back, it's already there. And then it says select a genome to use. We are using fruit fly DM6 because that is the data that I'm using. So come here, click, and then select your fruit fly. So this also lets us know on Galaxy, you can also run GoSeq if you have a human genome or mouse or zebrafish or rats. So those are things you need to also do. And of course, if you are using it on R, you can download the respective files or the respective packages to help you perform this kind of analysis. We will look at those ones in other videos. Okay, so for now, let's select our DM6 and then let's do this. Let's scroll down. Let's see what else we have. It says select gen ID format. It's ensemble. That one we already have it there. And then when you come here, go cellular components, biological process, molecular function. So these are the groups for the Go terms. So Go terms can either be cellular components or biological processes or molecular functions. So whatever Go term you select will be under one of these, right? So just take note of that. Okay, so let's get back. So we have selected all these and then we need to check something else. So let's go to output options. It says output top Go terms plots. So we have to do that here. So come to output options, click it, output top Go terms, select yes. The next one, extract the Dagents for the categories, go/Kirk terms. That one too is here. So we just select it. And then we come to run to. So you can click this or you can also click this to run it. So either of them should run it. So now, once we do that, we are going to find 3 outputs here. So what we have to do is to wait for Galaxy to process them so that the background turns green like this. And when it turns green like this, that means that everything is done. And then we can examine the outputs. So let's wait for Galaxy to complete its work. All right, so whilst we are waiting for the analysis to get done for us, let's explore the inputs again. I thought it would be good for beginners to also know what we are doing. All right, so these are the inputs. We have the gen IDs and lengths. I will open that. It's a TSV file. I'll just open it here. All right, so this is it. So we have the gen length. So I have them here. So I have the gen and then the length. So this one here, if you run feature counts to quantify your gens, to get a read count, let me just show you. This is an example of a feature count output. So if you open it, let me just open it. I think I already opened it. Let's see. So there's an example of a feature count output. So feature count also computes the gen length. Let me just enlarge it a bit. You can see the gen length here. So you can use that information. So you cut this and then you cut the gen ID like this, and then you merge them and then you get this, get something like this. So that's how we're able to get this information. And then the other inputs, this one here, let me just open it. Here's the gen ID, then the D status. So whether the gen was differentially expressed or not. So this one, to this and this, you get it from the results here. So this one was generated using the Seq. So there's the D results, the differential expression analysis results. We have it here. And so this gen, these are the gens. This is the column for the gens. And so you will have to set a threshold to determine if it's differentially expressed or not. So a threshold, you can use the Q. Let me just open it here. We have some stuff here. We have the p-value. We have the p-value. We have p-adjacent value. We have log2 full chain. So there are criteria you can use, but one of them is going to be the p-adjacent values. Or you can also check the galaxy tutorial here. It has an example of a threshold that you can use to determine if a gen is differentially expressed or not. So I think that one is here. Yeah, so it uses this one here. I think there's a p-adjacent value. Yeah. So if it's less than 0.05, it's differentially expressed. And so if it's differentially expressed, then you have true against the name of the gen. Let's just check it here. Let's use this one here. It's true. If it's not less than 0.05, then it's false. Like, let's check here. Like this one, for example. All right, it says false. So you look at the threshold you want to use, and then you can get this table. So you can use R or Python or any other text processing tool to get this. And then once you have the two inputs, then you can run GoSeq. All right, so you can use R or you can use Galaxy, like what I'm doing. All right. So again, if you have a data set and you want to perform this kind of analysis, you can always contact me. Or if you want to learn how to do it, you can always contact me. You can have a session together, and then I can explain a couple of things to you. All right. So let's get back to our Galaxy tutorial here. So this is it. So we have the outputs. So we have 3 outputs. All right. So let's just check the Galaxy page here. So with the Galaxy page, it gives us a brief explanation. We have a table which has this information, and that one can be found here. Let's just check. So this one here, that's it. So you can download it. Let me just put it somewhere here. Yeah, I can put it in this same directory. So this is the GoSeq run category list using this method. This tabular. We will just maintain it as it is. And then I'll go and check it out. This is it. So I'll open it with a spreadsheet software. So I'll open it now, and I have the information. So here you can also do some statistics, but that's something I'm going to cover in another tutorial. But you can use that to answer the questions that has been given here as well. So just try that, because it's basically the same data I'm using. So try that to answer the questions here. So you have the table here. These are the descriptions for the columns. So you can just use that and then make inferences, because the information, everything you need is here. So just use that and then answer these questions. We also have a plot, which is here. So that one can be found here. This is it. There's the plot. So when you click it, you can just download it. It's a PDF file. So you'll see something like this. All right. And you can even compare with what has been given here on the Galaxy page. So there's it. So there's it. So you can just compare and then just use that to also answer the questions. All right. And we also have this. So this is a PDF. You can download it. Mine is already downloaded. So I can just open that. Let me just open it on my PC as well. So I'll check it out. So there is it. So I can open it. So there's it. So you can open it. It's a PDF. So you can just open it on your PC. And also just check it out. And then we also have this other one, the Dagent for the categories. You can also download it and check it out. So Dagent. And save it. Yeah, that has been saved. You can check it out and also open it. This is it. Yeah, so you can just check and see the Go terms. So the Go terms, you can even check them online as well. Let's get back to the Galaxy tutorial page. So you can use the information you have to answer the questions here. So it says how many Go terms are represented with adjusted p-value less than 0.05. So you can just check that and then answer it here. So to do that, you have to come to this section here and then just filter. We have the p-values and stuff. So you can just go to filter and then get what you want. So for that one, I will leave it for you. But when I'm doing the R version of the tutorial, we are going to look at how to use the R code to explore this and then get some more information. All right, so that's something we are going to do with R. And please take note that you may not get the exact same number or the exact same values as we see in the answers, but you'll get something closer. All right, so just take note of that. Okay, so that's it. So let me just repeat again that you may not get the exact same value or values that you see, but you get something close, and then that will help you to get started. All right. So some of these things, the differences may occur due to maybe differences in software versions or maybe a slight difference in what the Galaxy system has been changed to. Normally with the Galaxy platform, you have updates and stuff going on. So there are a number of factors. So some of these things, if they are different, you can investigate further to find out what happened, and then that will help you to get things done. Or you can also contact the Galaxy developers and then just see how stuff go. But in general, it should be fine. And some of these tools are also a bit stochastic, so that will lead to a slight difference in what you have. But nevertheless, we have been able to reproduce this tutorial, so that's fine. We can also look at the plot, which is here. I think that one I showed it earlier, but there's it. So you can just check, and then we have ours also here, which is there's a slight difference, but again, the slight difference is coming because the results we got is slightly different. So obviously, the plot we have here, the figure we'll have here will be a bit different from what has been given on this tutorial, so you need to take note of that. But this is the procedure for performing a Go enrichment analysis using the Go-Seq package on Galaxy, and so that should be fine. Now in the next tutorial, I'm also going to show you how to perform this statistics. It's a simple one, but I just thought I'll make a separate video on that so that we look at how to analyze the output for Go-Seq. So you will look at this one here, you will discuss it, use some R commands to get the output here, and then that'll be it. So that's something I'm going to cover in another tutorial. So just make sure to check the description box. If the video is available, the link will be there. All right, so that's how we do it. So again, if you want some additional help on how to perform this kind of analysis, you can always reach out to me and then we can work something out. All right, so that's how I bring this tutorial to an end. And in the next tutorial, I made mention we'll do an R tutorial. And then we are also going to look at the Keck Part 2 analysis using GoSeq. So I'm currently working on that. So I'll also share that. So the GoSeq package for the Go Enrichment analysis. This is the Galaxy tutorial episode. I'm also going to work on the R itself, where you use your local R to do that. And the same applies to the Keck Part 2 analysis. So we will do everything on Galaxy. And we're also going to do everything on R so that you can have a bit of both worlds. All right. So that's all I have for this tutorial. For those who also want to support the channel, there are links in the description that you can use to buy me a copy. So I will see you in the next video.